All right, so we just finished talking about glycolysis. Now remember, this is where glucose is broken down into two pyruvates occurring in the cytoplasm and having two NADHs, so two hydrogens removed from our C6H12O6 molecule. Now the products go into the mitochondria, so this is our pyruvate right here. It's a 3-carbon structure. It moves into the mitochondrial matrix. I'm only going to demonstrate this with one pyruvate molecule just due to lack of size, so room to fit the molecule. So one pyruvate moves into the mitochondrial matrix. At this point, one of the hydrogens is going to be removed by NAD. Of course, this now becomes NADH. This NADH will now move on to the first membrane protein channel for electron transport, which we will get into later. After this, a carbon is removed from our pyruvate. This is now given off as our first carbon dioxide molecule, which of course will be exhaled. We now call this structure acetyl-CoA. This will combine with a 4-carbon structure in the mitochondrial matrix. The 4-carbon structure is oxaloacetate. And when that happens, then the acetyl-CoA will go through an 8-step process of being broken down in the citric acid cycle. Now, during the citric acid cycle breakdown, the acetyl-CoA will have the final 4 hydrogens removed from the C6H12O6. Now, 3 of these are going to be NADH, and we'll go ahead and use this one again. 3 of these are going to be NADH, and then 1 is going to be FADH2. Now, this becomes significant in the electron transport chain, which we will get into later. So these, just like all the other NADHs, will take their electrons down into the electron transport chain. The significance, or the significant difference between NADH and FADH2 is going to be where they're going to hand off that hydrogen and electron. It's in a slightly different location on the electron transport chain, which we'll do later. Now, the final 2 carbons are going to be broken off of the acetyl-CoA molecule. Of course, you will be exhaling that as your main waste product. 1 ATP molecule is created in the citric acid cycle from the acetyl-CoA molecule, and of course that will go wherever it is needed. So, just to kind of wrap it up here, the acetyl-CoA molecule, which is 1 2-carbon structure here, the acetyl-CoA molecule, has 4 hydrogens removed, 3 of which end up on NADH, and 1 ends up as FADH2. The 3-carbon structure, pyruvate, which is the end product of glycolysis, gives up 1 hydrogen molecule, and it gives it up to the NADH. That's 5 hydrogens, C6H12O6. And during glycolysis, this half also gave up 1 hydrogen to NADH as well. That's 6 of our hydrogens. So if we double that, there's your 12, C6H12O6. And our carbon structure of our glucose, which becomes pyruvate, which then turns into acetyl-CoA, is completely eliminated at the end of the citric acid cycle as carbon dioxide. And then all the hydrogens from that sugar molecule will end up going to the electron transport chain. So ends the oxidation of pyruvate and the citric acid cycle. Next up is following the hydrogens and how they're going to hand off, is following the NADHs and how they're going to hand off the hydrogens in the electron transport chain.